Here we are finally in Chapter 10. We've covered everything in the inside of a cell, from atoms to molecules to cell structure, and now we're going to get into cell reproduction, and this and the next chapter will get us set up for doing actual genetic problems. But first we have to understand cell division before we could get into that. This lecture will also set you up nicely for lab this week, which is mitosis. These images show mitosis in early development of a sea urchin. We had a fertilized egg that splits into the first division where we get 2 cells. We continue, and now we see that the cells are no longer all the same size. There's an uneven division of the cytoplasm. And eventually, through repeated cell divisions called mitosis, we end up with the fully formed animal. And we can see that it's a sea urchin in these images, but with the first 2 pictures, that could be any animal that early in division. Briefly going to mention prokaryotic cells here. The second half of this chapter, which will be a separate lecture, will go into the details of prokaryotic cell division. But they have a single circular chromosome, and they do not have a membrane around that chromosome, and so they can divide by binary fission. Where the chromosome gets duplicated, the cell gets slightly bigger, and it splits into 2 identical cells. There are other ways that DNA is duplicated. This image shows 2 prokaryotic cells that are joined by a pilus that was originally only on 1 cell. It sticks to the second cell and creates a channel. And then the plasmid, which is a separate piece of DNA from the chromosome, can be duplicated and passed to that other cell. That is just genetic exchange, but then when this cell undergoes binary fission, it will duplicate both its chromosome and that plasmid and pass it on to its offspring. The rest of this is going to be dedicated to what happens when eukaryotic cells divide. This is an image of a karyotype. We've seen Karyo before. That is the suffix in eukaryote or prokaryote. And that karyo means the center or a kernel. And in the terms of eukaryotic cells, it is the nucleus. So eukaryote cells have a true nucleus. Prokaryote cells have exist before the nucleus came into play. This is an image of actual chromosomes that have been stained with fluorescent dyes. This was the nucleus that was burst open. And then this digital photograph can be manipulated so that we can actually arrange the chromosomes into their pairs. The subtitle here tells us that this is a human cell. All humans should have 23 pairs of chromosomes, and a pair means that 1 chromosome came from the maternal parent and the other chromosome in that pair came from the paternal parent. And they carry information for the same traits in the same position all the way down along that chromosome. And here, to aid with visualization, they are stained with different fluorescent colors. There obviously are not fluorescent colors in our cells. The 23 pairs, each pair has unique information on it. This has 2 X chromosomes, so it says that this is a female human somatic cell. Somatic just means that this cell was not an egg cell. Somatic cells are all the cells in our body other than the ones that are for reproduction, for sexual reproduction. And typically, a human female will have those 2 X chromosomes. And if this were a human male, there would be 1 X and 1 Y chromosome. All the other chromosomes are similar. They vary in their size. There's this little notch in the middle. That is where there's proteins at the center called the centromere, and that region is going to be important for how we organize the chromosomes during cell division. We can see that some chromosomes have an equal amount of DNA on either side of that centromere. Others have very little on one side versus the other. Just depends on which chromosome that is. Now, this doesn't mean that if you don't have this exact 23 pairs of chromosomes that you're not human. There are obviously variations in that, but any variation in the number of chromosomes, or if there's extra pieces or missing pieces on a chromosome, that will almost always have some sort of physical and health effect on that human. I included a few slides here about biological sex, simply because we had an image that showed chromosomes with 2 X's and said that was a human female. And that's generally true. Chromosomal sex or biological sex is not quite as simple as we've thought in the past. I've provided some images here along with a link. This is a link to a Radiolab podcast called Gonads and XY. It has to do with, typically, in a female, there are 2 X chromosomes, and this one has shading on it because there is something we'll get into with meiosis called random X inactivation. In any human that has 2 X chromosomes, only one of them is going to be active in that cell. In persons that have 1 X and 1 Y chromosome, in the typical pattern, we call that male, and they have this gene on the Y chromosome called the sex-determining region Y, or SRY gene. So there are abnormal cases where the SRY gene is either located where it shouldn't be or it's missing. So typically there should be in an XY male, at least a genetic male, an X chromosome and a Y chromosome, and on the top little piece of the Y chromosome is that SRY gene. However, during meiosis, separate chapter, there can be a mistake made when the chromosomes are shuffled, and you might have 2 X chromosomes, but if one of those X's has that SRY gene, then this person physically appears male, has complete, typically expected male secondary sexual characteristics. In addition, we could have someone with a normal X chromosome and a Y chromosome where that SRY gene is missing, and chromosomally they are an XY person, but would develop physically with secondary sexual characteristics of a female. And that doesn't even get into gender identity, separate thing. So a karyotype could tell you there are 2 X chromosomes, but may not be specific enough to tell you are they 2 normal X chromosomes or is one of them carrying an SRY gene. A karyotype could tell you the person has an X and a Y chromosome, but the karyotype would not be specific enough to tell you does that Y chromosome have the SRY gene. Getting into the details of mitosis. The DNA in our cells, when they are not dividing, is very relaxed. It's still condensed so that it fits inside of our cells, but not as condensed as it will have to be in order to do mitosis. So if we look at DNA as its double helix, it does wind around histone proteins to form nucleosomes, and so this here would be only this tiny little part where DNA is wrapped around the proteins. And if we zoom out on that, each of these little gold coils are the histone proteins. The blue is the DNA wrapped around it, and then those can twist and compact further so that we get chromatin, and eventually we will see a duplicated chromosome. So what I have tried to draw over here is in our duplicated chromosome. These little gold dots are representing that centromere, the middle of the chromosome, or a position on that chromosome with proteins. So if we have one chromosome with its centromere, and before we can do cell division, all of the chromosomes have to duplicate. And when they do, they are held together by their centromeres that fuse together. We have two identical, now they're called chromatids. And those two identical chromatids that are stuck together at their centromere is somehow still one chromosome. This was one of the hardest things for me to wrap my brain around, that I can see double the amount of information. Why is that not two chromosomes? Well, it's not considered two chromosomes because we don't have any new information. When everything goes right during this phase of duplicating the chromosomes, the new one should be completely identical to the first one. And it's there and stuck at its centromere only until we complete cell division and it pops apart. And then one of those chromatids goes to one cell and one goes to the other, and now they're separated and are back to being called chromosomes. So in order to know how many chromosomes are present, you count the centromeres. The number of centromeres will tell you how many chromosomes are present. Move me back to the corner. The cell cycle is a description of sort of the life cycle of a single cell. And it's either in a state that's not resting, just metabolizing but not dividing, or it may receive a signal that tells it it is time and necessary to divide. We have this tiny little orange pie slice here is the mitotic phase, the actual cell division phase. The entire rest of the cell cycle is called interphase. So we have separate portions of interphase called G1, S, and G2 for the first growth phase, the synthesis phase, and the second growth phase. It's called interphase because it is the phase that is in between the time to divide. So time-wise, the cell spends the vast majority of its time in interphase, even if it is an actively dividing cell. The actual cell division, the mitosis, is a very, very tiny part of the overall cell cycle. So different things happen at these separate pieces of interphase. Initially, we have cell growth. So the cell does have to get a little bit bigger before it divides. Otherwise, the resulting cells will continually shrink every time cell division occurs. So it gets a little bigger. And there's also going to be checkpoints that we'll see in the second lecture on regulation of this. that double checks, did the cell grow? Do we have enough nutrients? Synthesis, we do have to duplicate the DNA. If we do not duplicate the DNA, then the resulting cell will not have enough genetic information. And not only the DNA gets copied, all the other organelles and everything else the cell will need to create two cells has to be duplicated. The second growth stage is making sure it's big enough, making sure there's enough nutrients, double checking that the DNA did duplicate the way it was supposed to, and then there will be signals that tell the cell we now have everything we need in order to divide and actually do mitosis. Not all cells will divide. Our brain cells, it's not that they never divide, but we have very little additional cell division going on there. So many of those are in what's called G0, which means they are permanently in G1, which is normal cell function, all the regular metabolism that cells have to do. This very tiny picture goes through the stages of mitosis and tells us what's happening at each of those stages. This book uses prophase and prometaphase. I will sometimes just say prophase to encompass both of those. There are a few different, more advanced stages that happen in prometaphase. But essentially, and I'm going to show some larger images and go into the details, we have to condense those chromosomes, and they have to be organized. They will get pushed to the middle. They will get pulled apart. The nuclear membrane will form around each cluster of the chromosomes, and then the cytoplasm will split. Now let's go into each of those stages separately. There is a slightly bigger picture of all of those. In prophase, the key things that happen is the nuclear membrane starts to dissolve. The chromosomes start to condense, and if we were looking at this on a slide, they would start to be visible to us. If we look at cells that are not actively dividing, we can see the nucleus if we add a stain, but it's just sort of diffuse and all the same color. When we get into prophase, the chromosomes have already duplicated. The chromosomes duplicate during interphase. That G1, S, and G2 have to be completed before we get to this stage where we see the chromosomes condensing in prophase. These little spiders on the corners are the centrosomes. Too many words beginning with C-E-N-T, but centrosomes. That is where the cell generates microtubules. We've seen microtubules when we talked about eukaryotic cell structure. They are part of the cytoskeleton, but they are also these little railroad tracks that are going to organize and push and pull the chromosomes during mitosis. So the microtubules are called spindle fibers. And the last thing that's happening during prophase is the nucleolus is no longer visible, that region inside the nucleus where ribosomes are made. As we're wrapping up prophase and moving into metaphase, the prometaphase, our nuclear membrane is almost gone. Our chromosomes are super condensed, and we have kinetochores, which are this cluster of proteins at the centromere, and that is where the microtubule fibers will attach. And some microtubule fibers attach at the kinetochore from each side, and others do not. And those are there to push the edges of the cell apart as we move further and further through mitosis. And those centrosomes are moving so that they're on opposite poles from each other. So now we've got our chromosomes attached to fibers. Our membrane of our nucleus is just about gone. It will be gone by the time we reach the next stage. And now we're ready to organize our chromosomes. And that happens in metaphase. The fibers that are attached at the kinetochore push the chromosomes to the middle of the cell. That is a region called the metaphase plate. There's not actually a physical structure there, it's just a position. These chromosomes are still as their sister chromatids, right? This is counting the centromeres, 1, 2, 3, 4. Still only 4 chromosomes. The nuclear membrane is gone. And we are ready and organized to be able to distribute our chromosomes in the next stage. In our little fluorescent image down here, we can see just how many microtubule fibers there are and all of the chromosomes smushed to the middle. In anaphase, the chromatids come apart, and in telophase, T-E-L-O means far apart. We've gotten them as far away as possible. So there were proteins that were binding those chromatids together. They break down. The microtubules that are attached to the chromatids get shorter, pull them apart. And the non-kinetochore fibers, the ones that are not attached to the kinetochore, get longer, and that's what's pushing the edges of the cell apart. That's creating the space we need so that in telophase we have released the spindle fibers. They're no longer attached to the chromosomes. The nucleus starts to form around each cluster of chromosomes, and this bright little dot there is the nucleolus beginning to reform inside of this nucleus. So we temporarily have one cell. It's got 2 nuclei in it. If we did not proceed, this would not be a good cell. We don't want 2 nuclei. So we have to correct my typing. I have telophase up here. I'll fix that. This is the end of mitosis, but now we have to have the cytokinesis, where we actually divide the cytoplasm. We complete having a perfect nuclear membrane around each of our chromosome clusters. The chromosomes are now diffuse once again, and now are called chromatin, and the cell will completely split and be 2 separate, genetically identical cells. Every single cell in our bodies started out from one fertilized egg cell that has undergone billions upon trillions of mitotic cell divisions. And so the DNA in the nucleus of one of our skin cells should be absolutely genetically identical to the DNA in one of our liver cells, or one of our heart cells, or any other cell. Because each of those cells is derived from an original fertilized egg that divided by mitosis. In animal cells, this split is accomplished by what's called a cleavage furrow, where actin cytoskeleton components pinch and divide the cytoplasm apart. In plant cells, we get a cell plate, which are vesicles that contain cellulose, so that we have both a cell membrane and a cell wall that separates the daughter cells. So in prometaphase, we have that kinetochore, which is at the centromeric region, and the mitotic spindle microtubules attach. There is one centromere, so even though there are 2 dangly bits of DNA, this is one chromosome. The cytokinesis, move me out of the way. In an animal cell, it is made of actin, which is the component in our microfilaments component of our cytoskeleton, and that ring contracts and pinches off the cytoplasm to create two cells. In a plant cell, as we're getting to anaphase, we get vesicles from the Golgi that are membrane-enclosed sacs, and inside that sac is cellulose. And cellulose is the component of plant cell walls. As the nuclear membrane starts to form, the vesicles containing cellulose fuse, and because the outside of the vesicle is phospholipid membrane and the inside is cellulose, we have phospholipid membrane and cellulose around each cell. So we do have two new plant cells, each of which has its own perfect nucleus, each of which has a cell membrane, and each of which has a cell wall. In the mitosis lab, you will be looking at both whitefish blastula, which is a very early stage in embryonic animal development, and it's pretty much a solid mass of cells. And we can scan across that. This cross-section of the whitefish blastula has been treated with a dye that specifically stains the chromosomes. And you'll be able to scan and look for the various stages of mitosis in the animal cells. We will also look at onion root tip to look at plant cell mitosis. The plant cells are a little easier to see. They're bigger because of their cell walls. Their chromosomes are a little larger, but we can see the mitosis happening in both cell types. We will begin next time with this slide, which is cell cycle regulation. The lab this week is just going through mitosis. The lab next week will be meiosis, which will be next week's lecture. So I will put this where I always do in our lecture discussion module, but I will also put this lecture into your lab notebooks so you don't have to go two places if you need a refresher as you're working through the mitosis exercise.